Welcome to the Novogene end of year review. This is our final webinar of 2020. Before I hand you over to your host for today, I'd like to thank you all for joining us on this webinar, and particularly thank those that have joined us for some of our other webinars throughout 2020. Whilst the move to running webinars was somewhat forced on us by circumstances, it's proved to be a great opportunity to share some awesome science direct to your desktop. A bit of housekeeping before we start. The webinar with a 10-minute question and answer session at the end. Please submit your questions via the Q&A button. You'll see at the bottom of your screen. You'll notice that you now have the ability to upvote questions that you like by clicking the thumbs up symbol next to the question. We will endeavour to answer your questions live. If we can't fit them all in, we'll get back to you by email after the webinar. Talking of which, we will share the slide deck and recording via email. You can expect this on Friday. So without further ado, I would like to introduce you to your host for the day, our European Sales Director, Yuan Chen. Yuan, over to you.
Hello, good morning. Welcome, everyone. This is our end of year review for our webinar series for 2020. We thank you for your time and your support throughout the year. And today we would like to have a short summary of what we have done this year. 2020 is a special year, I think, for us and for most of the researchers. We are hit by the pandemic. And despite all these difficulties, Novogene has managed to deliver some awesome science. In the past, we were delivering our scientific stories and seminars through the offline platform, for example, by going to conferences and giving seminar talks in university. However, this year we moved all of these activities online, and it has proven to be quite useful for our research partners. And we were hoping that next year we can continue this and we can bring you more scientific stories. So at Novogene, we have this platform to share with you the most recent advance in technology and the most advanced development in methods. And we can share with you case studies, whether it's a newly published paper or whether it's an ongoing study, excuse me, whether it's a PhD defense. We have this platform that allows researchers to share their scientific discoveries. So by doing that, we have a webinar series, we have a user forum, and we have a case study showcases. So I would like to show you, for example, what we have done in 2020. We have hosted a total of 14 webinars in 3 languages. We have over almost 2,000 attendees attended these webinars. And the topic of the webinar really covers a great topic, a series of topics, all the way from RNA sequencing to human genome sequencing, to the bioinformatics supports, to microbial solutions, to epigenomic solutions, and to some of the de novo assembly studies. These series of webinars, they are available online on our website. You are welcome to go to our official websites at Novogene Webinar Hub. You will find the list of existing webinars from throughout the year. they're all on demand. You just need to click, and then the video, the recorded video, can be played at any time. We will constantly update this list. So if you can register at this page, you will be automatically updated if we have any new webinar come out. We all accept the webinar. We also made a user forum. This is a 2-day forum. We invited our clients. We invited interested customers. And we also invited speakers from both scientific community and from industry to give an overview of what the technology will move towards and what kind of applications can be done for the scientific research. We had the user forum separated into plant and animal topic and human research topic, and over 400 attendees attended, and these videos are also online. You can check anytime. Except that we also had a case study to broadcast some of the very recent scientific discoveries. This one case study, which is also online, is talking about a modern Egyptian genome. So Novogene and our client, our collaborator in University of Lübeck, Dr. Holger Busch, together published a Nature communication paper on the first comprehensive Egyptian reference genome. And we also delved into what are the significance and what can be the future applications to facilitate precision medicine. So we have planned in next year, 2020, even more interesting seminars and webinars. If you are interested in sharing your study with your peers, please feel free to contact us. We will be more than happy to host a webinar together. Or if you're interested in more advanced technology, more advanced sequencing methods and applications, please sign up to our webinar series and you will get notification with the newest updates. So with that, I would like to introduce to today's speaker, Dr. Wei Li. She's our senior product manager, and she will introduce some of the most recent publications that Novogene has published in 2020. So, Dr. Li, it's your turn.
Well, thank you for the introduction, Yuan Yuan. If you can stop sharing, I'm going to share my slides. Okay, can you all see my slides? Well, I hope so. Well, good morning, everyone. First of all, I'd like to say it is my great pleasure to give you the publication review for Novogene this year. Well, 2020 has been a little bit tough to almost everyone. However, we still achieved a lot in the scientific research area. And in this webinar, let's just look back to what has been done in this year and how Novogene support different researchers to achieve their goals. Before we move to the review, I'd like to share the comment published by Dr. Richard Gibbs in Nature Reviews. He is one of the earliest contributors of Human Genome Project. He says the success of Human Genome Project should be measured by how this project transformed the rules of research the way of practicing biological discovery and the ubiquitous digitization of biological science. The success of Human Genome Project is also reflected by the publication record of Novogene. From 2011, when Novogene was first founded, we have been contributed or involved in more than 550 papers, and the total impact factor is about 4,000. Those projects are researches about human, animal, plant, and microbes. Just in this year, by October 2020, we have supported almost 80 papers with the total impact factor around 700. All of those great works have been published in top journals, including Nature, Science, Genome Biology, and others. To meet such diverse requests, we not only provide standard services, such as DNA sequencing for human, plant, animal, and microbes. We also provide transcriptome sequencing for coding RNA, non-coding RNA, isoform, and single cells, and epigenome sequencing, including whole genome bisulfite sequencing, ChIP-seq, and RIP-seq. We also provide services for pre-made libraries and customized analysis. So in the future slides, I will share some leading-edge advances in different topics. First, let's check the DNA sequencing part. As listed here, it includes the de novo sequencing for different species, resequencing, and microbial community sequencing. While de novo sequencing is applied whenever you don't have a good reference genome. So it is like to complete a puzzle like showing here, when you have no idea what it should look like. Before you see this 2D picture, we have to try thousands of times to see what makes sense. To complete this puzzle, well, in the research part, that means we will generate the good de novo assembling and annotation for the reference genome. We have to overcome some challenges by only using the illumina data or the short reads, such as when you have repeat regions or highly heterozygote regions or regions with extremely high or low GC bias. We have to combine illumina data and also long reads generated by the PacBio platform or nanopore platforms. So by combining both short reads and long reads, it will allow us to generate the good or high quality assembling and annotation for the reference genome. Usually, the de novo sequencing is quite common for newly discovered species. However, we can also use the de novo sequencing for the well-studied species as well, such as this example. In September, as Yuan mentioned, the team from University of Lübeck, Dr. Bush led the team and published a paper in the Nature Communications. So this is the EgyptRef project, and they first successfully constructed the de novo reference genome, for the Egyptian population. We already have the Frontline Genomics webinar available in our website. So if you are interested in the details, feel free to use the link to check more, to check our recordings. And in this one, this project was initiated in late December of 2017. As I mentioned, for the de novo project, we usually combine the data from Illumina platform, which generates the short reads, and also the long reads generated either by PacBio or nanopore. So that will give us to address the complexity of the genome. And in total, we generated more than 800 gigabytes of data with the overall genome coverage about 270. With those data, the team tried to assemble the genome with different pipelines, the two pipelines listed in the middle. And they also generate the final meta-assembly on the base of the primary pipeline, the primary assembly. With those, I'd like to summarize the very basic idea about to perform the de novo sequencing projects. So our basic idea or our primary objective is to deliver the high quality reference genome and the annotation, or simply saying we try to complete the puzzle as listed here. And the top pictures are showing the previous publications about the de novo projects has been supported by Novogene. With this good reference, we now can move to other projects, such as you are interested in the differences discovered in the materials that you have. And with that, you can compare your materials versus the reference. So this process will be the whole genome resequencing or whole genome sequencing. As shown in the right circles plot, so this is showing the reference genome in the very outer circle, and we can also identify listed variants as shown here in the middle circles, including the single nucleotide variants, insertion or deletion, copy number variants, and structural variants. For human project, it really based on the regions you are interested in. We have different options, such as if you are interested in the variants in the whole genome scale or in the whole genome level, and you want to identify the structural variants, so the whole genome sequencing will be a good choice for you. However, you may be just interested in the whole exome region or a specific group of genes. Then you may choose the whole exome sequencing or targeted region sequencing. With that, it will be, well, very difficult for us to identify the structural variation or even the copy number variation. However, those two methods will deliver much higher sequencing depth as shown here. So back to the EgyptRef project. Once we got the reference genome successfully assembled, we can map our other materials or other data, such as the data they collected from other Egyptian individuals, and identify the single nucleotide variants, indels, and also structural variants as shown in the pie chart. Well, if you check the Venn diagram in the right, we can tell that some of those SNPs in the Egyptian population is actually included in the larger data set identified in the European populations. This is because most of the current genetic studies are performing based on the European population as a reference in our data set. And those differences shown in this Venn diagram remind us that some of the population or specific population is actually underrepresented in our current data set. And this project, I hope it can give you more idea about how we can perform de novo sequencing for human species in the post-human genome project era. In addition to the population scale study, we can also use the resequencing method to study a specific cancer or disease, as the example shown in this one. So this team is trying to study the multiple gastric cancer, and which presents in multiple sites of the patient's stomach. And they want to study the carcinogenesis of the multiple gastric cancer. To achieve this objective, they collected the samples from tumor and also normal tissues, and they used the whole exome sequencing to identify the mutations. from the data, they found out in this one, the first line, 8 out of 12 patients are carrying the splice site deletion, one types of the mutations in the MSH2 gene. And this gene actually participates in the repair during DNA replication. So with this kind of discovery, they kind of suggesting this deletion may be a great or maybe an essential contributor to the multiple gastric cancer. Therefore, all of those results may help us to better understand the disease and how the cancer came up in different patients. Well, the resequencing can also be performed on other species, for example, the plant species. In this project, the team actually sequenced more than 1,000 rice accessions to study the hydrolysis patterns. Like showing or like summarizing here. So in total, the team generated almost 4 terabytes data with an average mapping rate about 99% and an average genome coverage depth about 17x. Based on the SNPs they identified from the data, they constructed the phylogenetic trees like showing in the right. That will tell us the relationship or the evolutionary relationship between different accessions or different lines. Also, based on the SNP information, they also calculated the frequencies of parental variation differences, like shown in the below. So this is a more direct method to study the rice hydrolysis. And if they got higher frequency, like the gene showing here, RF4 gene, that means this loci is more associated with this kind of phenotypes or the hydrolysis phenotypes. And actually this gene involved in the fertility of the rice lines or rice accessions, which also give us very valuable information for the future rice breeding. So in summary, no matter you perform the whole genome sequencing in what species such as human, plant, animal, or individual microbes, our basic idea is to identify the variants. And all of those variant information will support you to further study the population-specific characteristics or to better understand cancer, disease, or some physical or some phenotypes. And also the phylogenetic relationships between or within a population. Except the individual whole genome sequencing, we also offer the microbial community sequencing. So this could be achieved by either the amplicon-based metagenomics or the shotgun metagenomics. In both methods, we can identify the community components through species annotation. However, for the shotgun metagenomics, in addition to the species annotation, we can also perform the functional annotation for genes identified in the metagenome. And with that, it will help us better understand the functions or the pathways have been carried by this community. So we have been working on different types of samples as listed here to achieve different objectives. Currently, we are supporting a 10K metagenome project in Netherlands. This project is led by University Medical Center Groningen, and they will collect the feces samples from 10K participants to study the potential role of gut flora in promoting good health and preventing disease. However, this project is still going on, and we hope we will have very exciting results in the very near future. So to show you more example about the microbial community sequencing, I'd like to show you this paper has been published in this year. In this paper, they are trying to understand the gut microbial population under the influenza infection. So they got two objectives. First, to identify the specific anti-influenza gut microbes, and the second, to better understand the survival mechanisms. To achieve those objectives, they set up the experiment like this. So they prepare the pathogen-free mice and infect them with virus. After the infection, they collected the feces from either survival mice or dead mice. They also performed the fecal microbiota transplantation into new pathogen-free mice, and they track their survival rate after the transplantation as shown in those curves. To better understand the population in gut, they performed the first 16S amplicon-based metagenomics after the infection, and they track their change at different stages after the infection. It is quite clear that we see a decrease of the green group and an increase of the blue group in the dead samples or dead mass when you compare to other survival group or control groups. They also performed the shotgun metagenomic sequencing at different stages. So here I just show you some of the figures they published in the paper. In the PICO-A analysis, they can cluster the samples based on their community similarity. And you can see a clear shift from those control or survival samples to the dead samples, which clustered in the very big red circle. And in the middle, it is showing the results from the Life-C analysis. In this one, you can identify representative species in survival group versus the dead group. And as I mentioned, in the metagenomic analysis, in addition to identify the species components, you can also analyze the gene functions or pathways which are carried by the community. So the right picture is showing the results for the pathway study. In different group, either survival or dead. So they identify the enriched pathways listed here, carried by either group or with the presence or absence of those representative species. So all of those data will help us better understand how those species can survive after the virus infection. With mentioned method, so our microbial community sequencing may help you discover or track the community components change, identify representative species or essential pathways in your interested community. And based on the different types of samples or objectives you have, so all of our both amplicon sequencing or metagenomic sequencing could be used for researches in ecology, human health, or food industry. In addition to the DNA sequencing, we also offer the transcriptome sequencing and the epigenome sequencing. Both transcriptome sequencing and epigenome sequencing are pretty popular in the research area as well, because they can help us better understand the regulation mechanism of the gene expression. In the transcriptome sequencing, we are trying to target different types of RNA as listed here. So we have surveys for coding RNA and non-coding RNA. And if you are interested in the interaction or the regulatory networks among multiple types of RNAs as shown on the left, you can also choose our whole transcriptome sequencing service. So in this one, you can sequence all above types of RNAs in a single run. Well, in total RNA, actually the ribosome RNA is taking a huge proportion of the total mass. So to enrich different types of RNAs, we will prepare different types of libraries. For example, for the messenger RNA, as they all have the poly-A tail, we can use the poly-A enrichment method to prepare the library. While for the long non-coding RNA, since they don't have the poly-A for us to capture, so we usually will use the ribosome removal method to enrich them efficiently. And transcriptome sequencing is a very efficient way to track or to capture the transcriptome profile in samples. And in this example, they focus on the interaction mechanisms between plant and the pathogen by using the RNA-seq. So the team infected the plant with the fungus, and they collected the samples at different time points and tracked their transcriptome change. The heat map showing here is a commonly used method to present the upregulated or downregulated genes identified in the RNA-seq data. Well, in addition to the differential expression analysis, we also provide analysis such as the enrichment analysis, identification of the SNP or indel, and alternative splicing analysis. For some of the projects, such as human or mice project, it is possible for us to do the fusion gene analysis as well. If you are interested in all of those details for the analysis contents or pipelines, you can always go back to our website and check the on-demand webinar recordings. Generally, all of those analyses will help you identify the novel transcripts or genes, variants, or discover the transcript abundance or track them. And with the non-coding RNA-seq or the whole transcriptome RNA-seq, so you can perform the regulatory network study as well. So that will be very useful for you to move further for the host or pathogen interactions. Well, for the epigenome services, in this one, they also involved a lot in the regulatory of the gene expression. So it really depends on what kind of information you are interested in, such as you want to study the methylation level of the DNA, or you want to check the interactions between different micromolecules, such as RNA versus protein or DNA versus protein. So we have different choices for you as well. For the methylation study, you can choose the whole genome bisulfite sequencing. While for the histone modification or the DNA protein interaction, we have ChIP-seq, and we also have the RIP-seq for the interactions between RNA and proteins. Whole genome bisulfite sequencing is a very traditional way to study the methylation level of the genome. And usually, we will follow this step to construct the library. So we start with the DNA fragmentation. We ligate the adapters on both ends, and we perform the bisulfite conversion, and then move down to the library sequencing. So in the bisulfite conversion, unmethylated C will be converted to U and further to T in the sequencing, while the methylated C will remain as C, as shown here in the sequencing part. However, in this step, as shown on the left, bisulfite conversion will introduce a lot of DNA damage. With that, we actually have a lot of broken libraries, which are not very useful for us, or we cannot collect enough data for the analysis. So that will ask us to prepare a lot of DNA input to meet our data requirement. To address this problem, we propose this method, the PBAT method, post-bisulfite adapter tagging. Still, we perform the DNA fragmentation first, but after this, we switch the bisulfite conversion and the adapter ligation. So that means we perform the bisulfite conversion first, which is followed by the adapter ligation. And with that, it will allow us to use the DNA input as much as possible. And if you check the numbers showing here, we bring down the DNA input from 2.5 microgram to 100 nanogram, showing on the right for genomic DNAs. With this method, we have supported a lot of research by using limit DNA input or DNA extracted from FFPE samples or from the cell-free DNA samples. And in this one, it's showing an example about how you can use the methylation sequencing to achieve your research objective. In this project, Novogene supported the library preparation and sequencing. And the objective is to explore the relationship between the methylation level versus the gene abundance. As shown in this figure, you can check the gene abundance shows in blue versus the methylation level shows in black. And it is clear to see the negative correlation relationship between the DNA methylation level versus the gene abundance. For the IP-seq, this one, we mostly focus on the interaction of the biological macromolecules. So basically, in those type of services, researchers usually design or select the protein of interest. And with this protein, they can interact with either RNA or DNA and to form the complex. And the specific antibody can capture out the complex with banded DNA or RNA. And with that, after the immunoprecipitation, so we can release the DNA and RNA and move down to the library preparation steps. So by using those types of methods, we can first to study the transcriptome targeting region or the histone modification in genome, or the second, we can study the RNA stability and structure or the microRNA targeting regions by using the Resequencing. This paper is showing you the example of the Resequencing. So in this one, we actually helped with both RNA-seq and the Resequencing. Well, this is quite special Resequencing as it's just used for the methylated RNA study. So they designed a specific antibody to only capture the methylated RNAs. And with that, you can see the modification in RNA with different methylation levels. So for the IP-seq projects, we usually need to prepare an input, which is the background before the immunoprecipitation, and also the immunoprecipitated RNA or DNA. And by comparing the IP versus the input or the background, we can check which region has been enriched significantly, or we call this as peak calling. So you will see those peaks across your samples, and those will be the most interesting regions you want to look further. So as I mentioned, transcriptome will be a very good way for you to quick scan the transcript and their associated abundance, while the epigenome will be a good way for you to better understand the regulatory mechanisms, such as the methylation level in DNA and how my RNA has been modified with different methylations. Well, we can also check the potential interactions between DNA or RNA versus proteins. So by combining those information or data, that will help us better understand how my gene expression has been regulated in the samples under different treatments. So from previous examples, I believe that you saw a lot of researchers have been using multiple omics or using different multiple solutions to address or to reach their objectives. And here is another example that we have contributed or supported in this year. So this one is a very huge project, and it has been led by the Chinese. It has been led by the Chinese Academy of Science. So this is a project about the Chinese prostate cancer genome and epigenome atlas. In this one, they kind of use almost all the services we provided, including the whole genome sequencing, whole transcriptome sequencing, and DNA methylation sequencing. They collect all the information from more than 200 samples. And in this figure, we summarized the results from this atlas, including the physical characteristics such as the age of the participants, and also the genetic information listed in the bottom, such as the copy number variants or structure variants identified by the whole genome sequencing, fusion gene, or the mutations in coding RNA and non-coding RNAs identified by the whole transcriptome sequencing, and also the DNA methylation information identified by the whole genome bisulfite sequencing. So this atlas or this map actually provide us very valuable information to better understand the prostate cancer in Chinese population. So basically, Novogene is providing end-to-end service solutions or solutions to all different types of researches. So from the extraction to the bioinformatic analysis, you can feel free to contact us and join us at any touch point we listed here. And we'd like to deliver you the high quality data, publication-ready analysis, and personalized service for all of your projects. So before we move to the Q&A section, I'd like to say I wish you a peace and lovely Christmas, as this is quite special year for everyone, and we hope we can visit you in very soon, and we can talk about more details of your research projects. So with that, we can move down to the Q&A section.
Hello, okay, thank you very much, Wei. It's excellent talk. We have several questions come in from the audience. We can use this time to go through it one by one. If you still have questions, you can use the time to tap into the Q&A. We will answer them live. So the first question is from Enrico. The question is, if we do whole genome bisulfite sequencing in my sample, do I have to do a whole genome sequencing on the side? Because these techniques are only complementary.
Well, if you have the reference genome already available, and you can just perform the whole genome bisulfite sequencing for your samples, and with the analysis pipeline ready, we can map your race back to the reference genome and to check the methylation levels at the single nucleotide resolution. So if you are still interested in other information, like other variants associated or showing up at the same position of the methylated site, you may use the whole genome sequencing as well to give you a joint analysis or more information for you to check in the future.
Yeah, I can also add on to that. We do have customers asking whole genome bisulfite sequencing together with whole genome sequencing. for the purpose, for example, the genome bisulfite sequencing is actually to understand the gene expression level changes, because even though the modification is on genome, the methylation itself mostly regulates the transcription process rather than the genome, for example, SNP or indels. So we do have customer combining bisulfite sequencing with genome sequencing. On the other hand, we also have customer combining genome bisulfite sequencing with RNA sequencing. So both, depending on what is your research goal and what kind of scientific question you would like to answer. We also have customers only doing bisulfite sequencing. It's all up to the design of the study. Yes. I hope that answers your question. If not, you can feel free to write emails to europe@novogene.com. Our local sales representative will contact you and will give you a call and will answer more details questions. The second question we have from Helena is for ChIP-seq. What is the best sequencing depth for fruit fly? Is 10 million reads sufficient?
Well, for the ChIP-seq, we do have recommendations, but still based on your research objectives for the by using ChIP-seq. So usually for the I think it's for the histone modification, we use minimum 20 million reads to check that. And for the transcription factor targeting regions, we usually use minimum 40 million reads. So it really depends on the research purpose, and those are kind of the very general recommendation for overall projects. But if you do have special requests for the, I think it's the fruit fly, we can still work together to see which sequencing depth is a good one for your project.
Mm-hmm. And one good thing about the service from Novogene is, especially for the cases where you cannot decide the optimal sequencing depth, you always have the options to add additional depths afterwards. So you can start with ordering 10 million reads. We will keep the libraries for how long? 2 months or 3 months. Then after your analysis for the first round, you realize it's not deep enough, then you can add another 10 million or 20 million. We have this option, and I think this will be very helpful for this kind of questions.
Yeah.
Next question from Nina. As for the PBAT method you referred, is there any limitation for the species? What is the minimum amount you can accept?
For the PBAT method, usually all the kind of the method using the bisulfite conversion is only applied to eukaryotic species, because the prekaryotic species is carrying different patterns of the methylation. So basically, for majority of the eukaryotic species, you can go ahead to try the PBAT method. And I think that the second amount will be the minimum amount. So as I listed here, so as I listed in my previous slides, so we, such as for the cell-free DNA, it's just a 20 nanogram. And I think our lab also achieved in even lower input. So if you have very limited input, we can just feel free to contact us, and we are happy to try for you to see if this is possible for us to perform the PBAT method for your methylation study. But only one limitation for the analysis part. So if you have, such as you have multiple samples and you really want to compare between samples or between groups, so once you have any sample is under the PBAT method. You have to use the PBAT for all of your samples to make sure when you perform the comparison, we only have the treatment as the only different, we only have the treatment as the factor to change the methylation, but not the library preparation pipeline to introduce another factor to affect to introduce more bias in your result.
Mm-hmm. And the PBAT method is a library for the whole whole genome bisulfite sequencing. It's actually one of the strongest strengths that Novogene have. We have references we can give to you upon request that recently finished a huge batch of human cfDNA with the input around 10 nanogram to 20 nanogram per sample. The bisulfite conversion rates are all over 98%, and the mapping rate is also over 95 to 98%. So this is extraordinary good result. So when you're working with the bisulfite treatment, mostly the mapping rate will be low, but with our methods, the mapping rate are so good. So please drop us a message if you have related project. We will be much happy to give you more references. Another question from Anne Karin. For the RIPSeq experiment, how much RNA is required? Does the RNA needs to be isolated in a special way? Do you offer the full service from RNA? Estimated cost per sample, this we can drop you a message later. And how many biological replicates per treatment do you recommend?
There will be a lot of small questions, but let's address one by one. So for the input request, so we do have the sample requirements details in our that we can send over to you in the after the webinar. But for the RIPSeq, I think we also have the low input method available, and I think the input could be lowered down, maybe to even lower to 1 to 10 nanogram. So it is very useful for it is very efficient for us to construct the low input library by using the RIPSeq materials. And for the RNA isolation part, so because for those types of IP-seq experiments, we strongly recommend you to use the fresh sample, and we usually only accept the IP RNA or DNA and together with the input. So for the RNA isolation, we can provide you some recommendations or method, and also we can send it over to you after the webinar. So usually it should be quite similar to the common RNA extraction, like the rhizo or any kit used by any kit listed in the commercial company. And again, for the full service for the RIPSeq, unfortunately, we cannot do that because we only accept the IP RNA and the input RNA as starting materials. But everything from that point, we can take care of the library preparation and also the analysis. So for the cost, our sales will contact you after the webinar. And the replicates, so basically for all of those RNA-seq or other biological experiments, we usually recommend minimum 3 replicates, because with that, it will give us the possibility to identify any outliers and to check if my experiment is repeatable, if others want to follow the same protocol. So we usually recommend minimum 3 replicates. But if you really has very limited samples, well, we can discuss the details and see the situation, the detailed situation that you have, and to see how we can address if you can only provide fewer or such as 2 replicates. So it is doable, but may introduce more false positive. So that's why we would like you to start with 3 replicates.
Okay. There is also a follow-up question from Anne-Catherine about the topic, what we just mentioned, that you can add additional sequencings. So the question is, if we add more sequencing depths to a sample to achieve higher depths, what about run-to-run variations? For the transcriptomic study, with poly-A variant, would it be okay to generate the cDNA twice, or is it too big a changes to that will introduce by that?
For this question, I think it really depends, or it's case by case. But usually, we will recommend you to preserve the library, because if it is in something like DNA format, it will be more stable. But if you have to re-prepare everything from the RNA, so sometimes you may include some internal control or internal spiking to check the batch-to-batch variation. So that will also give you the possibility, if there is any variance between batches, you can normalize or remove that variance. But except that, we always keep the same protocol as much as possible, and we do include something like a correlation analysis to see if the data from different batches can cluster together, or they have very similar results by using the same input in the data set. So again, it's really case by case, but we do have different recommendations or suggestions for you to better address the batch-to-batch variation.
Mm-hmm. And I think on this topic, what we normally do as a routine is we kept all libraries. So whether this is a poly-A messenger RNA library, or it is a whole exome sequencing library, we kept the library in our lab for 3 months, or I have to check the exact date, but you have a period of time that you can analyze the data. So in case you realize the depth is not enough, you can contact our project manager to say that for this sample, I need this amount of extra data. So we will pull out the exactly same library that has been sequenced before and load them onto the sequencer again to add additional data. So normally we would not start from the beginning. So we would not start with library prep. We would just pull out the libraries if there are any remaining. So sometimes we use up, but if there's remaining library left, we will just run that library again on the sequencer. So this way, the bias can be really, really low. And of course, you can run Pearson correlations to check whether there's batch effect. Okay, there is a last question from Ibrahim about can we get a personalized gene panel for targeted sequencing for cfDNA? Oh, this is a very particular question. Yes.
Well, for the customized panel design, we do have technical support to help you or to support you to perform the capture kit design. And for the panel, well, we really need to double check the details of your project, because if it is a clinical related project, we have to check if we are okay to perform the panel sequencing based on the regions you are interested in. But if it is only for research services, we definitely can support you to finish the kit design. And for the genes or for the regions that you are interested in.
Okay, our time are running up for today, and thank you so much for joining us today, and also thank you for your support throughout the year. We are wishing for Merry Christmas, and we will see you again next year at our webinar series. Thank you for your time.
Thank you.
And see you
Merry
next year.
Christmas.